Okay, so welcome back for the scientific session. So now we'll move on to the science, and I'll be happy to share you some of the work what we have been doing with respect to monocytes, where multiple technologies are being used to understand the mechanisms of monocytes and how they contribute to the disease mechanism, particularly in the field of tuberculosis. So this is titled as Molecular Analysis of Monocyte Subsets. So before going to this, I think everyone should be aware of this tuberculosis, a rare-born communicable disease, which almost infects every part of the human body. And here, it's predominantly, mostly it's pulmonary when it affects the lungs, and the rest of the body part is called as an extrapulmonary. So India, it has a very high incidence of tuberculosis. So every third individual in this country is being infected with tuberculosis. And it's a matter of fact how they are being exposed or symptomized or clinically victimized. And these are all the common symptoms for having TB, either fever, fatigue, weight loss, persistent cough, cough in the blood, and night sweats. So when this happenings in the symptoms are being monitored over a period of time, then there are more chances that you are maybe deceased with tuberculosis. So when it comes to tuberculosis, clinically and microbiologically, there are two confirmatory stages. So when you see the bacilli in the sputum, it is mainly confirmed as tuberculosis. Similarly, when you see a chest X-ray being with a lesion, X-ray radiation, these two things will clearly say you are diagnosed with tuberculosis. But when it comes to the host immunology, there is no distinct or discriminate stages which can tell how the host response is being varied or how it is going to be different. So particularly, there is no distinction and definition in the concept of host immune response when it comes to the immune response or inflammatory reaction or even the mycobacterial burden. So these things, there is no definition or there is no pinpoint evidence to say at what stage of you and where you will be progressing to the disease and how you are containing the disease. So understanding the host immune response is very essential and crucial in order to understand both hosts as well as the pathogen's role in the disease. So this host path interaction is very, very important. So one analogy what I can say is, so when a thief or a robber who always does the crime and go into the society, the police goes and find the culprit. So this is a small analogy. But in human beings or within the human host, the mycobacteria is the robber. It does the culprit activity and goes and hide in the police station because the macrophages does the policing job, and these bacteria resides within the macrophages. So it is very hard to confine them and to control them. So when you understand this small concept, then you can see how efficient is the pathogen. So for a successful host response after infection of this mycobacteria, you need certain mechanisms. And these mechanisms are initiated with the innate and adaptive immune response, particularly which ends up in the interleukins. And these interleukins activate the monocytes, and monocytes will help them to clear from the system. This is towards the favor of the host. Whereas if the pathogen gets success due to the lack of the immune system, then you will be having a hyperinflammatory response. As a result, there is an imbalance in the cytokine which will help the pathogen to grow. And as a result, you will become susceptible for the disease and leading to the fatality of the disease. So here you can see that there are different stages. Like what you can see is latent TB. One is like a healthy contact, other one is latent TB. This latent TB is almost like 90% of the people in the society. When I say it is like 1 in 3 is infected latently. And these people, they are exposed to the bacilli, but they are contained by the immune system. So they don't have any symptoms for the disease. That is called latent TB. And among them, 5 to 10%, they convert into active TB. And this active TB is a disease where you show all symptoms, particularly this cough, cold, and blood in the cough. These things are called as active TB. And within this active TB, you have two types. One is drug sensitive and another one is drug resistant. So drug sensitive is a case where it can be very easily cured by giving a combination of 4 antibiotics for a period of 6 months or 4 months, and they respond to the antibiotics and then they get cleared. And successful pathogen, they enter into a drug resistant mode where some of these pathogens, they don't get cured because they develop resistance against these known antibiotics. So for them to combine, we need a second line antibiotic, which is around 12 to 13 antibiotics, which is needed to supplement the primary antibiotics. So they are called as a drug resistant tuberculosis. So what we need to look forward towards elimination of the disease by WHO is all they want to have a target which says the TB incidence rate has to be reduced to 50%. From the 1/3 population, if you can reduce it by 50%, it will lead to the elimination of the disease. And second thing is TB related deaths has to be reduced. So more than 75% is a target of TB reduced death, which we want to do. But that is not being happening right now, which is only happening around 5.9% at the current scenario. Then what is the catastrophic cost involved in the TB diagnosis or cost towards controlling the TB pattern? So that is also like we want to make sure that people don't spend their money for TB. So we want them to spend zero amount of money, but still for diagnosis, patients are spending 48% of their expenditure. So these are all the problems towards elimination of the tuberculosis program. And the other major problems are this undiagnosed LTB population. So population who are infected, they are roaming around the society without knowing that they are infected. So this population has to be identified and treated. Another one is this multi-drug resistance of the bacilli, that is also very important. And the lack of diagnostic labs with high sensitivity and specificity is also very important. And we need some specific technology to discriminate the latent TB, active TB, drug sensitive and drug resistant. So once you develop this kind of technology, it is very easy to diagnose and to treat. So we are working towards this aim, and the component what we are going to study is about the monocyte. And why we are going to study about this monocytes, the reason for the TB disease pathology, I will explain in few slides, and then we will go back to the science. So this is a small immunology behind TB infection. So I think biotechnology people, you are aware of some amount of immunology also, and some amount of pathogens, and which will be very interesting if you know how the mycobacteria evades immune system. And this is a small schematic diagram, which is an alveolar macrophage, where the TB bacilli goes through aerosol. It infects the epithelial cells. As a result, there is a dysfunction of epithelial cells and dislodgement of epithelial cells, which in turn produce lot of chemokines. These chemokines, they attract the monocytes. The monocytes from the bloodstream come inside, and I want to say the monocytes are the precursors of macrophages and dendritic cells. So as and when they come into the alveolar system, the monocyte gets converted to macrophages, And this macrophage has an ability to opsonize the bacteria inside and to clear it mechanisms. So when it is in the process of clearing, there is a battle between the bacilli and the macrophage. So during this process, a complete cascade of chemokines are being produced by the monocytes and macrophages, and these chemokines, they attract neutrophils. These neutrophils, in turn, they rather attract the T cells and B cells. When the T cells and B cells comes into the picture, the macrophages do an antigen presentation system with the CD4 T cells through an MHC class mechanism. So when it is being successfully broken up, the parts of the antigen presentation is being presented to the CD4 T cells through MHC class, class 2 and class 1, and then they will take the antibodies and then antigens presented, and then these 9 CD4 T cells get matured to Th1 type or Th2 or Th17 type, and once the maturation of this Th1 and Th17 cells, it is again interconnected to the dendritic cells, where the rest of the soluble antigens are also being picked up, and which helps the effector T cells to be either Th1 or Th17. So these Th1 and Th17 is a very effective pro-inflammatory cytokines, which has the potency to secrete more cytokines, and they can combat, and then they can have the potency to help the clearance of the bacilli within the macrophage. And later, there will be some combination of T cells and B cells, and then they produce this antigen-specific IgG and IgG for the absorbed antigens, and they will take care of the rest of the plasma cells which is being produced for MTB-specific antibodies. And once it is being cleared, there will be some dislodgement in the metallothioneins. As a result, there are multiple components here which will fix the repaired epithelial cell as well as the monocytes. So this is a small picture which is very evident in terms of containing immune disease mechanisms, where the bacilli is being handled successfully within the pathogen. So coming to the monocytes, we all know that monocytes have very peculiar, unique identities, which are mainly characterized by means of these markers, that is CD14 and CD14 and CD16. So normally these two markers are very essential, and this is universal across the human beings. Besides, there are multiple markers which can characterize the help of the monocytes. And what are the main roles? They are like, there are 3 subtypes of monocytes. One is the classical monocytes, intermediate monocytes, and non-classical monocytes. The classical monocytes, they are helpful in terms of phagocytosis. Whenever any foreign particle, particularly these intracellular pathogens, it comes, it can be bacteria or virus. It will opsonize and take it inside the macrophages, and they can be converting them and then process them and then clear the pathogen. So that is mainly the classical monocytes, which is almost like 70 to 80% of the monocytes. The intermediate monocytes are responsible for antigen presentation, which is having the CD14 plus as well as CD16 plus. They patrol, so basically they go to the site of infection or inflammation, and then they perform the patrolling behavior, and then they are very good in the terms of antigen presentation. Then another one is the patrolling behavior of the non-classical monocytes. These are the non-classical monocytes which they don't have the phagocytizing capacity, but they will be very helpful in going to the site of inflammation and infection and convert them into an effector monocytes, and they will helpful in the healing process of the infection or inflammation damage. So knowing the small role of these monocytes, what is the main objective of our study, which is all about understanding the phenotype and functional differences of these monocyte subsets along with the transcriptome analysis, and then how it is happening between the latent TB, active disease, and drug resistant inflammations, and also in some cases of disease relapse. So we know that monocytes can contribute effectively in all the stages of tuberculosis, and for which our biotechnology component or instrumentation or experimental design can help you to identify. So I am going to share you some of the advanced technologies which we can expose or we can imply to understand these monocyte subsets. So basically a lot of phenotypic studies as well as transcriptomic studies are being carried out starting from the last decade, but we have contributed that there is altered phenotype across the disease subjects. The phenotypes are being altered and there is a difference in the phenotypic component, particularly the secretion of TNF and the other molecules which are being expressed on the monocytes. These are getting difference and they are very unique in the different stages of disease. This is very well studied in autoimmune diseases and some of the cardiovascular disease, but for TB there is not has been done effectively. Similarly, the transcriptomics is also not being studied, so we wanted to see how the transcriptomal is one of the tool where we can study the function of the monocytes by means of reading the mRNA and miRNA. So this was also very well understood here. So our specific aims for the project was to study the phenotypically characterize any different monocyte subset across the TB patients and see how the surface marker expression is varied when compared to the normal healthy subjects. It's very simple. Then second one is to study the transcriptomic profile and also to see how these candidate biomarker genes can help you in diagnosis or in the therapeutic events. So this is our simple target for one of the proposal and for which we have been working. And we have collected the samples which includes these 4 categories. The healthy controls. These healthy controls are normal human beings who have less exposure to tuberculosis and there are no symptoms of exposure to tuberculosis. So they are being identified by means of IGRA release assay. Normally when a pathogen goes inside, you will release interferon gamma, which is very specific for MTB antigen. So normal healthy subjects, if we screen all this population, around 20 to 40% of the people will be positive for this assay, which means 40% of these individuals are exposed to Mycobacterium tuberculosis. And these people are being right now protected because your young immune system and the healthy diet can take care of the protection. But as you progress in terms of your aging, you are bound to have symptomatics which will lead to the disease symptoms. Then we have the drug sensitive and as well as the drug resistant tuberculosis. Yeah, so now for the phenotypic study, we have this kind of small instruments of hematology analyzer where we can have this collected the blood samples and then we can do the FACS analysis, and from the FACS analysis we can look for the various markers on the cell surface which are nothing but the glycoproteins are labeled, which is labeled as a CD4 and CD8 markers for regular CD4 T cells, whereas in the monocytes it is CD14 and 16. Along with it, lot of multiple CD markers are being characterized and they are used for the phenotypic analysis. So when we do that, we can see that there is a difference in the types of monocyte subsets across the category. And you can see there is a healthy and latent TB, they have low number of monocytes, whereas the active drug sensitive and drug resistant TB has an increased monocyte subsets. And this can be studied by means of monocyte to lymphocyte ratio. So monocytes is only an 8% component of lymphocytes. So when you have a percentage of monocyte to lymphocyte, there is an increased ratio, MLR ratio, which is seen in the infected or diseased state. Similarly, this elevated monocytes in active TB, how these elevated monocytes are being represented in disease conditions. So that also you can see it here. So these are the classical monocytes where you don't see any difference. Intermediate monocytes, you can see some trend in the increase of these two at DS and DRTB, whereas non-classical there is no difference at all. So there is only a difference in the intermediate monocyte subsets, which had a very thing in antigen presentation, and which is because of the load of the microbial burden within the disease state. And another important finding what we have seen is there is a diminished disease. As I said, the monocytes are capable of differentiating itself to monocytes, from monocytes to macrophages and to dendritic cells. The macrophage component is being enriched, but the dendritic cell component is always being reduced. And this dendritic cells reduction is considered as a dysfunction which leads to the disease state. And this is the multiple system like UMAP analysis, which is a bioinformatics, and also like a FACS analysis, where multiple markers of other than the CD416 expression is being measured across different samples and different molecular markers. So these UMAP markers can show that these monocyte markers are being activated, particularly this HLA-DR, CD64, CD86 are being very well activated. So they are more activated in the DS and DRTB when compared to the healthy and latent. And also, as I said, the differential expression of the dendritic cell markers are also downregulated. So this is the summary of the phenotypic study, where what we can see is there is an abundant increase in the ML ratio for the disease and infected, and they have a different activation system. Molecular and monocytes activation systems are being increased. An accumulation of CD positive subset in the inflammation, which have a more of inflammatory role rather than a pro-inflammatory conditions. And we can also see the dendritic cell population is being downregulated. And we can see the observed frequencies of monocyte and DC subsets are very trivial, and it's not very significant when it comes to DR and DS. And UMAP analysis also helpful in the differentiation of monocyte activation markers and also the DC markers in the DRTB when compared to DSTB. Yeah, so now as I said, these monocytes are being regulated intrinsically by some other interleukins. So the interleukins which plays a major role in differentiating these monocytes within the system, both as well as the infected as well as the non-inflammatory sites also. So we tend to measure the cytokines or chemokines, which will be in turn very helpful in regulating how the monocytes are being differentiated and what monocyte subsets are being projected or promoted. So for that purpose, from the blood serum, we calculated the cytokines. We measure the cytokines and chemokines, which can give a clue about which favors the differentiation of monocytes. So this is basically we want to see like whether, because here in the phenotypic study we couldn't see why there is a difference between DS and DR, and which is very trivial. So we want to know whether monocyte differentiation or circulation is being indirectly influenced by cytokine and chemokine in circulation. So for this is a very simple method. You take the blood, separate the plasma, and then we do some techniques like Luminex assays. These Luminex assays is very advanced technology in measuring these soluble analytes, which needs very minute amounts of blood. For example, like 50 to 20 microliters, 20 to 50 microliters of blood, we can measure an array of cytokines and chemokines. Normally by ELISA, you need at least 100 to 500 microliters of blood or plasma. Here it is only 20 to 50 microliters of blood plasma, where you can analyze multiple components of things. So here this is Luminex bead assays, and we analyze them by these techniques, like all these statistical techniques to measure how these cytokines and chemokines are very important and which one is very helpful as a biomarker. So this is the importance of TB pathogenesis within the cytokines. And this is one evidence, like among the 14-plex cytokines measured in the TB patients, how many of them are being increased and how many of them are reduced. So the panel here, what we show here on the left-hand side, all these cytokines were found to be increased in the DS and DR when compared to healthy and latent TB, which means there is more amount of inflammation, and these are all pro-inflammatory cytokines, particularly this interferon gamma, TNF alpha, 17 and IL-1A. So these are all the pro-inflammatory cytokines. As a result of the feedback mechanism, you also see IL-4, IL-10 and IL-6. IL-6 is again a pro-inflammatory cytokines. So all these cytokines are rampantly increased in the case of DS and DRTBs. And this IL-5, this is found to be reduced in the DS and DR. So this is basically, it is an extracellular pathogen. So when nematodes or worms are getting infected, you will have this IL-5, which is a typical Th2 type of cytokine. And here, like, we don't see the significant difference when it comes to IL-1 beta or IL-18 or IL-13. So we can see that only the pro-inflammatory mechanism or an anti-inflammatory mechanism is being subsequently elevated when it comes to the disease pattern. So here you can see how different it is. So here we do a heat map analysis and show you that how when compared, this is the healthy state where you don't have these kind of cytokine increase, but when compared to the healthy, latent, you can have this interferon gamma and interferon IL-2 being increased. And from this latent, when you go to progress to drug sensitive, you have additional cytokines being upregulated. And when you become a drug resistant, even more amount of cytokines are being expressed in your body. And this we want to know that whether this can be useful as a marker. And among the all cytokines, we could see that only this IL-17 exhibited a higher marker by means of ROC curve. So we can say that this is very good in terms of discriminating the different stages of the disease. And we can also say among the 14 cytokines, these 8 cytokines are very important in discriminating all the 4 disease patterns, like 1, 2, 3, 4 groups are being very well differentiated with this one, predominantly by IL-17, followed by the other cytokines. Yeah, this is another subgroup analysis where we say a combination of 2 or 3 or 4 can differentiate the different group of patients. Then this is a summary that IL-17 can have a better discrimination, and a combination of IL-17 along with 5 other cytokines can differentiate DRTB, and the future validation of the cytokine signatures have a larger cohort of multiple sites can be useful for a diagnosis of DS and DRTB. Similarly, we also carried out for chemokines. Chemokines, we had 10-plex, and you can see there is an increase in the chemokines on these aspects, and there are no response for selected chemokines. And these chemokines are very helpful in attracting the monocytes to the site of infection or inflammation. Similarly, you can see among the different healthy contacts and the latent TB, there are multiple increase of chemokines, particularly CXCL1, CXCL9 and CXCL10. So this is what is very important in bringing the monocytes to the site of infection and making them progressing to the disease. Yeah, so statistical method to say that these are very important. And this is a summary of what we have identified, that CXCL9, 10 and CXCL1 are found to be a better biomarker to discriminate the various stages of tuberculosis. So these are all about the phenotypic difference and intermediate phenotypic difference. Now we will go to the functional difference. And for this functional difference, we need to apply some of the biotechnology like mRNA and mRNA technologies. So by means of studying the mRNA and mRNA, we can try to understand the functions of the monocytes in a better manner. So monocyte RNA sequencing technology, we apply the whole genome sequencing, particularly by Illumina technology, where the entire RNA, mRNA is being isolated and which has been sequenced, and it has been allocated to the identified genes, and which will be very helpful in see towards the functional aspects of the monocytes. So here what we do is, like the same with the blood, we isolate the PBMCs And these PBMCs will be stained with the fluorochrome antibodies, and these antibodies will be subjected to the FACS, and we will try to sort only the monocytes. And these monocytes will be helpful in sorting with the help of CD14 and CD16 antibodies. And from the sorted monocytes, we do extract RNA, mRNA as well as miRNA. The mRNA is being subjected to the Illumina RNA sequencing, which is a sorted single cell sequencing. And for the nanostring technology, what we do is, this is carried out in the presence of amplification by means of cDNA technology. Here there is no amplification technology. It decodes or give a marker, digital marker for the mRNA, which is miRNA, which is really present within the plasma. And these thing analyzing, we can see the differential expression analysis among these two isolated mRNA and miRNA, and we see what are the target prediction sites for this miRNA and mRNA, and we look for what are the pathways which are being altered or functionally very essential to do that. So here when we do this across different stages, you can see in a disease condition, there are multiple genes are being upregulated when compared to the downregulation or differential regulation. So when it is upregulated, that means they have a differential in the function. It can either suppress a gene or it can activate a gene. So in order to identify how many genes are there and what are the unique genes or a common gene or a shared gene, we do this Venn diagram analysis and see how which are all the genes which are being in between the different category. And so you can see here the common genes, this 584 gene signature, 6 and some 19 and 576. These are all common between these two comparative stages like latent TB and healthy controls, DRTB and LTB, and DSTB and LTB, and DS and DR. So these are all the different comparisons. When we across look for the monocytes isolated from those patients, we can always say that there is a clear difference and there are common set of genes which are being activated and common set of genes which are, or unique set of genes which are being activated at different disease conditions. So this is the PCA analysis to show that they are very different, unique and different, and how, what are the genes which are differentially regulated in terms of the fold change expressions. And these are the pathway analysis, bioinformatic pathway analysis, that what kind of subjects or what kind of genes which are activated or differentially regulated and where they are being followed. So it is very clearly we can see they are mostly in the Th17 response, natural killer cell mediated toxicity, and then also you can see in the downstream of TCR signaling and the PD1 signaling, also like some of the mechanisms like apoptosis and FcR2 gamma mediated calcium mobilization, and also some of the T cell receptor mechanisms. So these are very essential functions of monocytes which communicate the innate immune function to the adaptive immune function. So as I said, this is the connecting link between these innate and adaptive system, and these are all the mechanisms which are being intricately regulated by the monocytes. So as a result, the disease is being progressed. And there are even some of the hub genes have been identified which is having the transcription factors. And these transcription factors, particularly the FOXP3 and this STAT4, these are all the very important transcription factor regulation which has the potency to translate the T cell mechanism as well as the B cell mechanisms. So when this transcription factor is being altered, the disease conditions gets changed automatically. Yeah, this is another thing where you can see this MYBL1 and also this FOXM1, and these are all the transcription factors which are being regulated by the monocytes. So here again this MYBL1 is again being regulated between the LD and latent. So there is intrinsic and common transcription factor which takes care of the differentiation process across the varied disease patterns. Yeah, so this is a summary of mRNA study what we have seen. So we have seen that the major pathways enriched, and both DR and DETB compared to the LTB, there is an active disease pattern. And some of the disease pathway mechanisms like IL-12 signaling, interferon type 1 signaling, and PD1, FcR gamma 1 calcium mobilization. These are all the major pathway systems which are being affected by the mRNAs isolated from the monocytes. And this is one of the common hub gene which is seen very important in differentiation. And we are now currently working on how this mRNA regulated by mRNA integration analysis. Next, for monocytes mRNA, we went to miRNA because the miRNA has the potency to go and regulate the synthesis of mRNA. So this is one of the regulatory mechanism which we want to see how monocytes miRNA has the potency to regulate the mRNAs, for which we apply this nanostring, which is a very simple thing, like even an amount of 10,000 sorted cells, and RNA or mRNA isolated from 10,000 cells is enough to do this technology. And it's a biomolecule analysis where there is no amplification. It will literally calculate the number of mRNA molecules, and it will be helpful in analysis of whether they are present in more or less. So as I want to tell you about this mRNA, how this miRNA is being contributed in TB pathways, and where are all the places where it has a regulatory mechanism. And these are all the various places. So you see that it is in the TLR signaling, ERK pathway, which is a signal transduction pathway. NF-kappa B is mostly for the inflammation, differentiation of monocytes, synthesis of cytokine, all inflammatory pathways. Autophagy is a process wherein the pathogen is being killed and derivated, and the apoptosis mechanism where the cells undergo changes with the inhibitory effect as well as the promoter effect, and also in the phagocytosis. So these are all the various functions of monocytes which in turn regulated by these identified mRNAs. And how these mRNAs are identified, these are being carried out by earlier human studies or studied in the murine model or in the cell lines. So what we have identified from these cell lines, the common genes which are common among these three studies, either by human studies or murine studies or cellular studies, have been identified, and we have synthesized what are the mRNAs which can contribute to the regulation of monocytes. So with this background, we also did this mRNA nanotechnology, and we have found out that as the disease progresses, there is a downregulation of mRNA. So there is mRNA we found to be activated and upregulated, but miRNA was found to be downregulated, which means they in turn have a regulation on the mRNAs. These are all the different miRNAs which are across the category. What are all the node identified miRNAs which is very responsible for them to differentiate the disease pattern. And here it is having a bidirectional response also. So between mRNA and miRNA there is a bidirectional response. In a common situation where it is healthy subjects, you can see there is a suppressed immune system when compared to LTB versus HC. The moment you get infected or exposed to the pathogen, there is an immune suppression. And this kind of immune suppression helps the bacilli to live within the human beings. But as the bacilli live inside the monocyte for a certain period, the activation phase starts place, and they can be helpful in the complete activated stages. So as the disease progresses, you can see more amount of activation is seen at the end stage disease, particularly the DRTB and DSTB. So all these miRNAs identified, they are all because of activation, and this activation is happening by means of downregulation of certain miRNAs. So basically what we have identified is a bidirectional response between the active disease when DRTB and DSTB when compared to LTB and healthy controls. This hyperimmune activation of DRTB group is compared to DSTB, and more clues are needed by means of studying the competitive endogenous RNA. That is a similar type of mRNA which can have a multiple regulated phenomenon with the miRNAs. So this should be studied, and we can study the exactly what is the exact function of each and every mRNA which is being regulated. So generally put a conclusion here, when you are exposed to the pathogen, you can see there is a increase in the monocytic mRNA, there is an increase in cytokine and chemokines, there is a downregulation in terms of monocytic mRNA, and all the rest there is no changes. And this is mainly because of the suppression of multiple immune response. Whereas in the case of active disease, when the disease progress, you can see the monocyte numbers goes up, intermediate population functions of these monocytes responsible for antigen presentation is being escalated, and the monocyte mRNA is again being enriched, and the cytokines and chemokines are being pumped up. As a result, you have an hyperinflammatory state. In addition, the monocyte mRNAs are being downregulated, the lymphocyte numbers are downregulated, the dendritic cells functions are being also downregulated. With the downregulation of all these components, there is an activation of multiple immune response within the thing, which is not good for the host to contain the disease. So sometimes if it's not being taken care, it remains fatal for the infected person. So with this, I would like to conclude, and I would like to thank. This part is one of my PhD presentation students' work. It's Pavitra. She is who carried out this work for her PhD, and I thank the governments, Tamil Nadu state government, and the financial support by DBT. Yeah. Thank you.
Thank you, sir. It was a highly knowledgeable session.
Yeah. So I'll be happy to have some questions from our students. Let me know if you understand, or if you let me know what you understand. Yeah, otherwise. Yes, sir.
You shared with the mRNA, correct? Some of the mRNA markers has been shared in the human and murine and other samples, correct? So if we take that 4 biomarkers, like we can try to cure it using that as a biomarker, or like we can develop a device or something like.
Yes, sir. Certainly the approach is to see what are all the common regulatory mRNAs, because some may be functional in the cellular models, some has been identified from the murine models, but in human studies we have identified. One example is like miRNA 126 and miRNA 144. We know these targets have exclusively in regulating the inflammation.
Okay.
So we want to know that whether this inflammation is again a component of infection. So here we have seen that this, as the disease progress from latency to activated and DS and DR, the inflammation component is also being going up. And this is being studied as we see a downregulation of that particular miRNA 144 and 126. Similarly, we want to use this as a diagnostic marker also.
Okay.
So in order, because there is no single diagnostic marker which can discriminate drug sensitive and drug resistant. Only by the end of the 6th month after taking a couple of drugs, you will come to know whether he is drug sensitive or resistant. So if you have this as a marker, it is easy to study by the end of 2nd month or 3rd month. By studying the miRNA downregulation, we can say that he is going towards resistance or not. So that is the approach we have taken from the other 3 studies. Yeah.
Thank you.
So I have a question. It was a wonderful presentation. Thank you, sir. You were talking about the molecular mechanism and about the drug resistance. You were talking about the first line of antibiotics and the second line of antibiotics. And what about the side effects of these antibiotics? And if they fail, can we go in for any natural way of therapeutic measure?
Yes, ma'am. So basically there is, we don't have any natural way of dealing things. It's only a matter of increasing the antibiotics. As we know that antibiotics have their own side effects. Basically it has effects on the GI tract and liver toxicity. This is the known side effects of having these antibiotics. When you add on to the second line drugs like linaclotide and fluvoxamine and fluoxamines, fluoroquinolones, you will have multiple components like heart Q rate of the heartbeat is being damaged, particularly when you have bedaquiline. You see that the heart Q rate is being altered. Then eye is one thing. So retinal damage is being more when you give ethinomide. So that is also another damage which is being caused exclusively because of the linaclotide and fluopan. Then you have thrombocytopenia and also liver damage and nephrotoxicity. So all these things are happening because we need these additional second line drugs to take care of the drug resistant pattern. And there is no other alternate method to take care of it for once it is identified as a drug resistance. It is a matter of selecting the choiceful antibiotic which is being aided by means of sequencing, whole genome sequencing, identifying a particular mutation, and you have to do a phenotypic DST also. Drug sensitivity matched with the mutation can suggest which kind of secondary thing we can give, and that can be helpful as a personalized medicine. So this is what we are aiming as a biotechnologist. So we want to sequence the bacilli isolated from the drug resistant case, see where exactly it is mutation is happening, you select the drug according to that, and then give them for a definite period, so that you can avoid the secondary effects or the side effects of the other antibiotics. Normally it is up to 14 antibiotics now. We want to minimize it from 4 to 6. That is the target of doing that part.
I understand that, sir. Can we go in for a cocktail where we mix the antibiotics with some natural compounds, and nowadays we have the metabolites from probiotics also suggested as a future therapy. So what do you say about it?
So probiotics can be of a future therapy which can help in taking care of the adverse events which is having because of first line antibiotic drugs. So we know that GI tract adverse events are being more when you administer these antibiotics. So giving more probiotics or more prebiotics can help them in taking the balance of gut microbiome. As a result, the adverse events can be less. But when it comes to the killing of the resistant pathogen, these natural products, they are not of that use. At least to them now, whatever we identified from a natural resources has not yet proved very efficient. So that is why we are still depending on the other antibiotics which are not commonly identified for mycobacteria. Even if it is working for other pathogen, we try to apply it for it and see whether it is being killed efficiently at an earlier stage. But a lot of people, they work for natural resources from algae and from marine sources. They are having it, but they fail to tackle the drug sensitive. So drug sensitive, when it comes to isoniazid and rifampicin, they work par with it. But when you go to second line drugs, none of these extracts are being efficient to handling it. So that is the drawback we are facing when it comes to the natural extraction procedure.
Thank you, sir.
So students, you don't have any questions, which means either you have understood everything or you have not understood anything. So I think at least you should know what are monocytes and what these monocytes are capable of differentiating. Monocytes can differentiate into macrophages and dendritic cells, and how they take care of antigen presentation.
Good morning, sir. Thank you for the lecture. So I just have this doubt. Rather than using antibiotics, are there any genes in the bacteria that we can trigger it to self-destruct itself?
So that is a very nice question of a biotechnologic perspective. So there are multiple mechanisms which is called, you should be aware of this CRISPR-Cas system, which is a genetically driven system where certain enzymes or certain pathways can be deactivated. So this is theoretically possible, and this is possible in a lab system. You isolate a bacilli and you develop this CRISPR-Cas system where a selective gene which is responsible for the survival can be deleted. As a result, the pathogen can be killed. So this happens only in the in vitro systems. If you want to happen in the human beings, where the pathogen resides within the monocytes, you should not target the pathogen. You should target the monocyte, because monocyte has the ability to either to contain it or to destroy it. So we are working towards it, and what we are aiming is the dysregulated mRNAs and miRNAs isolated from monocytes will lead a clue. As I said, when these miRNAs are targeted in future directly into the lung alveolar macrophages and monocytes, those infected monocytes can be killed. It can be wiped out. But the problem is the host immune system is entirely dependent on monocytes. In an experimental condition, there are some lipoglycans where you inject into the mice system. All the monocytes are being cleared within days of 7 to 14 days. The infection, the inflammation is completely wiped out. But when it comes after 14 days, when you expose to the same pathogen from outside, you are again going to have a same set of infection. So thinking from eliminating or destructing from the pathogen side, it will be wise to think how to eliminate or target to contain the bacilli within the host immune system. So we should, instead of targeting the bacilli, you should target the monocytes or macrophages. That will be very helpful. Yes. Please.
Good morning, sir. It was a nice presentation. As this whole genome sequence analysis are costly to do, can we train some AI for identifying these biomarkers?
See, AF works on identified database. Whole genome is needed to identify the database. Without the knowledge of whole genome sequencing and without the pinpointing where are the mutations, we cannot apply the knowledge in getting a data structure to identify exactly what it is. So I can say one particular gene is mutated. How do you put the AF to say whether this gene is mutated or not? You should put the entire sequence, ask the intelligence to see where it is mutated, then to let us know. So that is why we need the whole genome sequence data to screen by artificial intelligence. So when you have a large number of data obtained from the whole genome sequencing, without proper database or an artificial screening tool, it goes waste.
Yes.
Yeah.
Thank you, sir.
Thank you, sir.
Yeah. Thank you, ma'am. Thank you.
Now, we will begin the second session of the day. Now, I'd like to invite Mrs. Merlin Diana, Assistant Professor, Department of Biotechnology, to introduce our speaker, Dr. Deepan Kumar, Associate Professor, Department of Biotechnology, Vellore Institute of Technology, to the gathering. Please, ma'am.
A very good morning to one and all gathered here. Today we have the honor of hearing from a distinguished expert in the field of biomaterials. I deem it a pleasure in introducing Dr. Deepan Kumar, Associate Professor, Department of Biotechnology, Vellore Institute of Technology, Vellore. He obtained his bachelor's degree in Microbiology and Advanced Diploma in Biotechnology from Anja College, affiliated to Madurai Kamaraj University. And after that, he did his postgraduate degree at Vellore Institute of Technology, Vellore, and with a PhD degree in biochemistry from Yunnan University, South Korea, Dr. Deepan Kumar brings a wealth of knowledge and experience to our symposium. At present, he is working as an associate professor in the Department of Biotechnology, Vellore Institute of Technology. He has also got a rich research experience. He has published around 25 research papers in reputed journals with high impact factor and 626 citations. He has also got a H index of 15 and item index of 19. His areas of specialization include biomimetics, biomaterials, scaffolds, bioinks for 3D printing. He is also well versed in many biological techniques, to name a few: protein purification studies, ELISA, FPLC, circular dichroism, etc. He is also a member of Society for Biomaterials and Surfaces in Biomaterials Foundation. We are much privileged to have you here amongst us, sir, to share your expertise. At this juncture, I would also like to thank our head of the department, Dr. Jasmine, for giving me this opportunity. Without further ado, I request the eminent researcher and Professor Dr. Deepan Kumar to take over the session. Welcome, sir.
First of all, thank you for the management and then the Department of Biotechnology for giving me this nice opportunity to share my research experience. So from this, I wanted to give you some of the experience, not to detail my research, because I think you will get bored. So I'm just sharing the knowledge of the research, what I'm doing. So I just giving you need to give a spark from your title itself. I need to give a spark to at least know my knowledge of research, what in the area of biomaterials. Okay. So like in the morning, correct, as our vice principal said, we are already trying to spoil the environment by using different materials like that thing. So, but in order to avoid this, what we have to learn from the nature is we're trying to mimic the system. So we need to inspire from the system to synthesize the new materials. So from basically from my area of research is like bioadhesives. So all the time we have a curiosity that we're trying to make some kind of adhesives or the glues using the chemical materials. Like how the nature, in the underwater itself, for example, the green mussels and some of the marine creatures, they try to barnacles, they produce a kind of adhesives during the underwater. So that is our concept to trying to prepare our synthesis of the adhesives. So basically, our research is like kind of a 3 different areas. Like first we try to inspire from the material, like the nature, from the material. Like mostly we are working towards the protein-based material. First we characterize the material perspective, try to identify protein-based material. Then we go for the genomics to a proteomic study where we can isolate the whole like protein sequences or the functional genome, like to express the proteins, correct? And then downstream process, how we can replicate to produce it in the laboratory scale, like recombinantly express that protein, and then purification, then how we can make the biomaterials out of that from the lab. So this is our overall the functionalization of our lab. So this was the work already published from the group. So what we inspired from that work is, they take the muscle adhesive protein, because it is well known that underwater the muscle adhesive, the creatures, muscles, correct? They secrete some kind of a protein called muscle adhesive protein. It can easily undergo for attached to the substratum, any substratum. So they combined with the curly fibers. So it is well known for the self-assembly property. So they make a fusion protein. So with the curly fiber and then the muscle adhesive protein fusion together and recombinantly expressed in the lab. So then expressed after that, what they expect is with the self-assembly property and then we can easily go for modifying the muscle adhesive protein to have a adhesion property, correct? So what they did is they make the fusion protein and then using the enzyme, mushroom tyrosinase enzyme, to modify all the tyrosin residue to the DOPA for the muscle adhesive protein can adhere together. So they synthesized it. So when we think of this material, why we have to take two protein and fuse together? Instead of that, we can go for the protein that have a self-assembly property. That is our concept, that we take the saccharine protein, we wanted to directly take the protein and modify the residue for the adhesion function. So what is the saccharine protein? We call it as, this species is called as red devil in the seawater. Like for example, jumbo squid. So the species name is like Decidius gigas, mostly found in the Pacific coast of the US region, South Atlantic oceans. So it is a fast swimmer. It has two regions, this is the arm, tentacle, and this is the beak. So we, in our lab, we used to work on two different proteins. So from the beak also we are working on it, and this is from the tentacle. So mostly this now work is based on the tentacle. So when we see these tentacles, they have a teeth. The teeth is completely made up of, this is the teeth, it is completely made up of a proteinaceous material. Mainly glycine, tyrosine and histidine kind of combination. So what is the interesting factor is, so this protein looks like a kind of a block of polymer. It interactively became like this structure. You can see here, it is a block of polymer. So intrinsically, the beta sheet domain and then the amorphous domain, that is the random coil domain, interacted sequentially evenly. So when we see this protein, we inspired from that how it is naturally possible. And then when we see the material perspective, this is the material perspective, it can exhibit a modulus behavior, like strength of the thing is from 4.5 to 7 gigapascal. It is more than the synthetic polymers that is commercially available, like for example PMA, polystyrene, polyurethane or PLGA, whatever the polymers, correct, synthetic polymers. This within the range, it is above than the synthetic polymer. So why not we can use this as a good biomaterial for synthesizing the material. So that is our concept and we are trying to work on it. So how we are going to modify the protein? So this is the general dogma, correct? Like you know, from the DNA to RNA transcription and then RNA to the protein translation. So generally we are focusing here on the translation. So basically we have 20 different amino acids, we have 20 different tRNA and tRNA synthetase. So each tRNA and tRNA synthetase pair, it will go for amino acylate the specific amino acid. So each amino acid has a specific tRNA and tRNA synthetase. Once it has amino acylated or the charged residue, it will go to the mRNA region. Here only the mRNA region only cares about the ribosome, correct? Only it cares about the interaction site, sense codon and antisense codon. If it is matches, then the corresponding amino acid will go to the growing polypeptide chain and form a full length protein. So here it is a concept, we are going to mutate, only concentrate this residue, okay, mutating this. So how we are going in? Before going for it, why we have to go for choosing the unnatural amino acid expression? Because there are 20 different natural amino acids are there available. Why we have to go for it? Obviously, you know that each amino acid has a unique structure. For example, best example is proline. It has a 5-membered ring. If you wanted to replace the proline, there is no other alternative in the natural amino acid. So that is the reason we are going out of the box. So this is the codon wheel for the amino acids. That is the other unnatural amino acids that are already established to incorporate into the system. So there are two approaches for incorporating the unnatural amino acids. One is the residue specific incorporation, that we are going to utilize the residue codon for the amino acid. We don't need any other specific instrument. We need a host system and we need a specific unnatural amino acid and incorporate into the specific residue. In the case of the site-specific unnatural amino acid, we need a specific system. Like for example, we need a orthogonal tRNA and tRNA synthetase, and also we need a specific mutant site to incorporate at a particular site. So here we are mainly focusing only on the residue-specific incorporation system. So what is the residue-specific incorporation system? Like, we need an auxotrophic strain. Auxotrophic strain means basically it cannot synthesize any particular biocentric pathway inside the host machinery. So we utilize the host machinery. For example, if you are here, I am giving an example for the tyrosine. So like we need to express tyrosine analog means we have to choose tyrosine auxotroph. In biosynthetically, it cannot synthesize the tyrosine inside the host system. And another thing is, for this, we have to choose the amino acid. It has to be isoanalog structure of the particular amino acid. For example, tyrosine and dopa, it has an isoanalog structure. So that is the important. Then we are using the yeast host system like tRNA and tRNA synthetase. For example, I think I'm giving some example like for the hostelers, correct? We are expecting like Sundays we have to have a meal of biryani. But when you go to hostel, maybe it will not like a biryani. Like what you do, it will have like a tomato rice. What you will do initially, we try to, we mimic, like we try to adapt it, correct? So similar way, we are trying to utilize that system, like we misaminoacylate the yeast host system, correct? E. coli host system, correct? To recognize this unnatural amino acid as your natural amino acid. For example, we grow at a E. coli for a particular stage. So the unnatural amino acid cannot be act as a building block. We need to grow a bacteria, correct? At a particular stage, like log phase, lag and log phase. So until that, we supplemented with the medium that is with the tyrosine. Once it reaches growth, we don't need the essential amino acid. That is the tyrosine, we don't need it. We need to use dopa. So what we do, at that time we depleted the tyrosine from the medium and we add the dopa and we add the induction system to induce the protein synthesis. So during that time, what will happen? E. coli started to misaminoacylate. There is an absence of tyrosine, correct? So E. coli tRNA and tRNA synthetase, which started to, and wherever the tyrosine residue, correct? For example, wherever the tyrosine residues for the sense codons is there, it will replace the dopa amino acid into that region. So finally, what you got, full length protein, wherever the tyrosine codons are there, it will replace to the dopa. So that's why we get the protein. So after this, we wanted to, our aim is to check the adhesion property. So we wanted to check. So basic study, we wanted to utilize this QCM and then the FTIR. So QCM is quartz crystal microbalance. So there we have a quartz crystal. Based on that resonance energy, we try to isolate whether the protein is absorbed onto the metal or the surface, any surface. So here we use mica surface, SiO2 surface and titanium oxide surface. So basically, we flow the buffers initially to stabilize the thing. After that, after stabilizing the instrument, we calibrate and then we started to flow the protein. If the protein started to absorb onto the surface, whether the silicon dioxide or the titanium oxide surface, it will show the absorption property. Then after a while, we again started to purge the buffer, because if the protein is specifically absorbed or just non-specific binding. So if there is a non-specific binding, it's just, again it has to go back to the original state. So based from that, we can identify that both the protein, saccharin and dopa modified protein, shows the adsorption property. In this FTI study also, we wanted to see like the adsorption, correct? Basically exhibited two kind of behavior. One is liquid water absorption and then surface bound water. Liquid means like based on the liquid interactions, when it absorbs, or it may only displace the water and go for the surface absorption. We wanted to see. So from that, we tried to, in the FTIR analysis, we found that it exhibits saccharin, native saccharin, exhibit only liquid-like water absorption. In the case of, you can see that here, liquid-like water absorption only it can show. But in the case of saccharin modified protein, shows surface-bound absorption. After this, we wanted to study the surface force apparatus, like used for the adhesion measurement. So this is adhesion measurement used to go for a molecular level interaction, like micronewton level interactions. So basically, we exhibit two modes. One is asymmetric, another one is symmetric modes. So we have two hard wall cylinders. Basically, in the asymmetric mode, what we'll do is, in the mica surface, we coat one layer of protein. And then the cylinder used to come in contact and then started to pull each other. Like for example, if it is like this, the cylinder will come in contact and then started to pull each other. So if there is adhesion, like for example here, correct, the cylinder used to come in contact with the sample and then started to again pull each other. If there is a adhesion, correct, it used to measure using the force, the negative force. It shows the adhesion. Okay. So in the case of symmetric also, both walls has been coated with the protein. So we wanted to see how the adhesion behavior. But luckily or unluckily, we had both proteins shows the similar kind of adhesion properties. Like in fact, saccharin shows much more. Without modification, it shows much more adhesion. Then what we found out it was, yeah, you can see here, we see that saccharin and dopa and like both cases, saccharin shows much more adhesive than the modified with the dopa protein. Then what is the role of dopa? There is nothing, right? So from this study, we clearly understand that adsorption and adhesion property is totally different. Adsorption property means like whatever the molecule, it just adsorbed onto the surface or binds to the surface of the molecule. Adhesion means the molecules has to interact each other and has some kind of applies some force. So both properties are different. Both properties are different. So what we concluded from this study is all the research, correct? What we hypothesize or what we expect will not be go in that direction. It might be kind of an experiment. So research will guide you to like kind of this thing. So when we come across at that time, there is a lot of other studies also reporting that initially many people think that from the muscle adhesive protein, dopa is the only molecule moiety can be responsible for the adhesion. Not like that. From barnacles, there is no post-translation modification of dopa molecule is there. And also from the other structure that they are pi-pi interaction. Kind of interaction shows that some kind of molecular adhesion behavior. So when we come across from some of the studies, might be our molecule have hydrogen interaction. Hydrogen interaction. Well, that's what we expected. So after that, wanted to like compare with some other saccharin like adhesive molecule, commercially available molecule. For that, for making adhesion, we have to formulate our protein. We know that saccharin has an adhesive property, but now we wanted to formulate the protein. So this is from our previous lab study. So what they did the interaction with the like form kind of a cross links. So using the ammonium persulfate and then the ruthenium, using the photocross linker, they form kind of a hydrogel. So with that hydrogel, we can able to tune the mechanical property of the hydrogel. Like for example, it can go from the kilopascal to megapascal and gigapascal. We can able to tune the material. So it is very good material because when you wanted to, for the tissue engineering perspective, you wanted to form a material, you have to have a kind of elasticity behavior. You have to have different property behavior. For example, if you wanted to make bone, you have to be very stiff material. If you wanted to make cartilage tissues or something, you have to be very flexible in nature. So we have to have this kind of nature. But here what we did, they published the paper with the ruthenium and APS. It cannot be compatible with the cells. So we have to find an alternative method. So then when we came across, we find out to replace the APS and then the ruthenium, we use riboflavin to have the similar dityrosine cross-linking. So when we use this dityrosine cross-linking, this is our aim. So we wanted to make a hydrogel to have some kind of adhesive behavior for the wound healing study. So once we wanted to wound healing study, then we wanted to further evaluate the immune response, how the immune response. And then after that, immune response, we also wanted to try with some kind of artificial skin, like bioprinted skin, correct? We wanted to see that how the efficiency of the percoloration studies, how it will effectively involve for the wound healing effect. So with that study, we wanted to first formulate the protein and wanted to comparative to the commercial market, correct? So commercially available glue was fibrin glue, well known, established for the material for the adhesive behavior. So we take that fibrin glue as a comparative material to our material. What we do is, here is, we take the two slides and attaches the porcine skin onto the slide. After that, we tape our hydrogel material. So once after this, we trying to use the shear force to break the molecule. So how much it will resist the force of the skin. Like for example, how much it takes time to shear it, tear it, okay? So basically, we attaches the porcine skin like this and then apply the force to pull each other to break the molecule, okay? So when it compares to the fibrin glue, this is the fibrin glue, with the different concentration of the saccharin we formulated, it was so much more adhesive than the fibrin glues. Okay, now it is adhesive compared to the commercially available fibrin glue, it is much more adhesive. Then we have to see compatibility with the cells. So that's why we check the BCI, that is the blood clotting index. Blood clotting index and hemolysis study. So with the different kinds of the material, like we tried. So both of these cases, blood clotting index and then the hemolysis study, saccharin showed better compatibility compared to the fibrin glues. And then we check with the cell viability. Onto that, we seed it to the cells and check the cell viability. It's not working, I guess. Yeah. Okay. So when we seed with the cell viability with a different number of days and then using two different cells, it is completely viable using our saccharin material. So that's what we see with the different stain, and then we can able to see that it is compatible. Then finally, we wanted to see for the wound closure. So in vivo study, we started to we take the non-treatment sample and fibrin glue and saccharin directly protein itself and then cross-linked hydrogel. So comparative to the cross-linked method, correct, with other methods like fibrin glues and every method, our hydrogel shows a better wound closure compared with the other developed method. So now with this, we developed and patented the hydrogel. Now with that, what we wanted to continue is, like as I said before, we wanted to see the immune response in the mouse model, in vivo model. What are the immunological markers, how it will behave when the saccharin fibrogen glue was there. And then also we wanted to see the percoloration study, like artificial skin. We wanted to take the bioprinted skin and wanted to see how the saccharin material can efficiently pass the sample into the percoloration studies. That is what we wanted to try to do now. So this is, with that, we want to continue with that work. So next work is, what we do is similar adhesive, like we wanted to see the muscle adhesive protein. So this creature, correct, like this is their muscle, foot. So this muscle foot used to probe all the places. Wherever it likes to have attached the surface, it just raises some kind of a protein and attaches the plex. That is the protein called muscle adhesive foot protein. So this muscle adhesive foot protein, when they try to analyze, initially it was a tyrosine protein in the present, when it releases from that. When it go for the post-translation modification only, the tyrosine has been converted to the DOPA. So we wanted to see how the molecular level changes when we develop our protein, correct, to modify the DOPA, how it will changes that, like behavior. So this is what the protein, like from the foot, correct, it releases a 3 kind of protein, muscle adhesive protein, 3, 5 and 6. So this 3 and 5, correct, later only come for the adhesion. Only the 5 protein, why we are targeting 5 protein is, this is the first protein released from the foot and try to reach us to the substratum and displace all the water and try to form the plug to attach us. So this is the 5 protein is the mainly responsible protein for the adhesion. So we are targeting the PVP5. So the PVP5 means it is the species, Perna verde species. So that is why we are telling that. So when we trying to modify that protein, yeah. So when we trying to see that, this is the food I am saying. When it releases the plaques, this is the adhesive protein. Okay. So this is the structure for the tyrosine and dopa. It has a similar structure, correct? Only the hydroxyl group was added to the thing. So when we see also, when we see the protein adsorption behavior of tyrosine and dopa and adhesion property, it is also looks similar. Adsorption was good in the case of dopa, but in the case of adhesion property, when we tried with the AFM and surface force apparatus also, adhesion property was similar. Then we from the thing last experiment also, correct? When we are working on the bioadhesive, saccharin, it is also similar behavior. So when we try to see the differences between the structures, yeah. So this is like a study kind of conservation, we used to say. So you can see that the sample here, when we add the protein onto a specific buffer, it tries to form a cloudy in nature. Yes. So this is the dopa protein. Here it is, you can see. You can see here, it looks like kind of aggregation. So what is the difference between aggregation and then the conservation is, you can see the clearly liquid-liquid phase separation. Like when you add the two kind of a buffers or reagents, it has to have a kind of a phase separated molecule. When you take this molecule and observe under the microscope, it will look like clearly look like a droplet. Yeah, you can see here, it clearly look like kind of a droplet image. If it, in the case of tyrosine, correct, we are able to observe, this is the sample, like it get aggregated. So when we see this kind of phenomena, we have to characterize the protein in a different pH buffer. We have to draw a phase diagram. What kind of pH it starts to stimulate, the phase separates the sample. So when we see that, the dopa molecule, you can see here, pH 7.5 to 8, has a recorded high phase change, phase diagram has been drawn. So we wanted to see, and what is the use of the particle formation? Like we can anything, we can load it into the drugs or the whatever the drug chemicals, correct? Like for example, any therapeutic molecules, we can load it onto the particle, correct? So that we just load the green fluorescent protein here. Load the green fluorescent protein onto the particle and see it, okay? Yes. So when we see this is the NMR structure, NMR diagram, HNQC NMR. So you can see here, this is the dopa molecule and this is the tyrosine sample. You can clearly see here, in the pH 3.5, there is no much differences between the sample. When it started to record in the pH 7.5 and then the above pH, it started to show a different kind of a behavioral structure change. So what we expect it was, might be when the protein, correct, exudes from the marine organism, it might have an alkaline condition. That might trigger the protein to form the adhesion of the dopa, correct? All the protein condensed into a concentrated form and form the plaques. So that is what we are expecting and we are getting it. So all the micro droplets, correct? The protein has been formed, assembled in a unique structure and a condensed molecule packed into form a fibrous unit. That is the plaque. So that's what we found out from this study. So far with this we published the paper, but we cannot able to formulate the conditions. So now we are working on the formulating the behavioral to either for the drug delivery or even make for kind of a fibril phenomena. We are expecting a kind of a study. We are further studying the property of this protein. Okay. Apart from this, we are also working on some kind of other interesting phenomena. So did you ever see this marine creature? It looks like kind of a eelfish, correct? It's kind of look like a eelfish. The name of the fish is called hackfish. Hackfish. when you agitate or tickle the body of this hagfish, correct? It used to excrete or secrete outside, correct? Some kind of a protein molecule. Some kind of a protein molecule that is similar like our keratin. We call it as mucin. So when this mucin protein reacts with the water, salt water, it try to form kind of a extended, like 10,000 times extended to form like a jelly in structure. Jelly in structure. So, like there is an accident happened in the US traffic. So when they have a large containment shipment, they are moving the hagfish from one place to another place. They made accident. It is like kind of a mess happened in the traffic accident. So like for example, even this hagfish, correct, even can clog a person inside the seawater. So mostly this phenomena the hagfish used to exhibit to either to catch its prey or escape from the predator. So these two phenomena. So why we wanted to mimic this behavioral study is when I was in Singapore, Nanyang Technological University, we used to go for the conferences, correct? So there the army defense people, they also came there to, they wanted, maybe they display some of the product or they wanted to have some of the product, correct, for the army purposes. They display it. So among that time, they wanted to have this kind of a small, like a small material, packed in a material, they throw in the water, they can react with the water and swell like big, because they have a close border, shared border with the Malaysia. So if you see the map in after getting the independence of Singapore, correct, 1962, and see the map of now Singapore, they reclaim the land very fast. So like they take the shared border, even the shared border is now just 10 minutes within the sharing, like you can reach by swimming. Okay, so they wanted to clog a person or a small sheep or whatever the migrant from the illegal migrant, correct? They wanted to clog the persons in the sea. So that is the main concept. So when we try to mimic the material, we experience a difficulty that when you're trying to compromise swellability, the strength can be increased. When you wanted to compromise the strength, you can increase the swellability. We have to meet in a sweet spot. So we try to mimic and also we try to functionalize the material. We found out this material, like initially what we make is, we have two polymers, make a hybrid network, form a double network hydrogel. That is very strengthable, is very high. When you react it to the water, the swellability is very poor. But when you make with this protein, correct, it looks like kind of a missile protein. So what we try to make is, we make the protein, the N terminus is silk protein, from the silk fibrin protein, spider silk, and then C terminal is also from that, and in between these two, we make saccharin protein. Make it this protein like form kind of a missiles. So when we make this kind of a protein, correct, missiles, correct, when we tune using the pH, it try to form some fibers. So basically, double network hydrogel and then this protein infused together to form a hybrid polymer network. So where we achieve the sweet spot in between the, there is no compromisation between the strength and swellability. When there is a strength has been dropped down, we can achieve from the double network hydrogel. When there is a swellability drop down, we can achieve from the protein because it can easily absorb the water and hold it inside the gel. So we achieve it using this phenomena. This is one of the study. And then in our lab, another study is like a tissue engineering approach. So as I said, the saccharin can use to tune the material, correct? We can be able to tune the material for whatever the wanted to make engineering, like bone or the cartilage or the skeletal or the bone muscle fibers. So here we wanted to make the myotube formation using the skeletal myotube formation. So where you utilizing the physical cue as our saccharin material as a substratum using the hybrid network. And then using the biochemical cues as growth factors or the cytokines or the any factors like it can stimulate the cells to trigger to form the myotube formation. With that, we are trying to make like this kind of pattern surfaces. We try to engineer to form the skeletal myotube formation. So this project we are working on it so far. we able to achieve the earlier myogenesis because the tuning property and also whatever the printing method we used for the antibiotic and antibodies, correct? It is not that achievable. We are tuning for the material now here. Yeah, finally, this is the another one marine organism. We call it as a mantis shrimp or the peacock shrimp. So the name comes from their colors. So it has a very good property is they call it as it is a crustacean and lies under the seawater. It has an arm. You can see this is the arm. It can release their arm more than a bullet speed. It can easily hit their rocks or the shells of the crabs and then the sea shells, correct? Without damaging their arm. With the one breakage, it can break the cells. But when we see the material property, it is made up of organic material. This research was completely accidentally studied. Initially, our lab, we have a material scientist. They used to characterize the material property. We have an aquarium there. One day, accidentally, during the stress, during the annual maintenance of the air conditioner, correct? This species jumped out of the tank and died. When we see that, actually the arms, correct? Every 90 days or 200 days, you used to shed their arms and try to form a new glove. So, and during particular period, it will be under stress. At that time, it also has annual maintenance of the air conditioning. So due to the further stress, it jumped out and then died. So we cut the gloves and then we try to see that property. We found out there are some proteins present in the arms. So when we try to identify, extract the protein and give for the sequencing, it is completely a protein. Then what we find out that we kill the species at every stage and try to see whether the protein is present in all the stages. So normally the gloves, correct? It sheds the gloves. Within the 2 hours, it can able to form the new gloves. But it will be soft in nature. Within 24 hours, it will form matured gloves. So during this 24 hours, the protein is stay there. What we find out from that is the protein can act as a biomineralization of like what we tooth, correct? From the calcium, from the tooth, correct? Similar behavior, it can try to accumulate the hydroxyapatite. So what we want to find out it is that similar protein try to mimic the mineralization of the hydroxyapatite. We proved from this study that the similar protein we recombinantly expressed and we try to biomineralize the studies, mimic the similar condition of the marine organism. We found out that protein is the responsible protein for the biomineralization. So now we are working on this protein to make our tooth gum filling and also for the bone marrow transplantation or whatever for the thing, correct? Because it is very light made material. And also not only that, we also making some kind of a bulletproof vest for a new material from this. So overall from this, my research, what we try to do is we inspire from the nature or we try to accidentally come from that and we try to mimic the material from the thing. So this is what overall our research from this is study. So these are my professors and collaborators who worked in or contributed in our studies. Okay. Any other further questions you can ask. Thank you. Thank you for.
So it is very fantastic and interesting phenomenon of biomolecules and their analysis. We need to analyze and characterize these biomolecules, but end of the day, like where and how it will be applied in terms of benefits. So one thing is whether can you increase the adhesiveness of any protein. Can you manipulate the protein by sequencing or introducing some sequence of the peptides or amino acids to increase the adhesiveness of a desired protein? That is one interesting thing like I just observed. If you can do that, then multiple miracles can happen in medical field. For example, this vitamin C is one molecule which we goes and bind on the blood vessels as and when it gets damaged. Since we do not have this binding efficiency of this vitamin C to the vessels, the cholesterol molecules and plaques goes and deposit on it. So if you have an capability to absorb the vitamin C and make it as an adhesive and plaster it on the vessels, the clots, plaques will not happen. This is theoretically I just observed from it. So all I want to know is whether you can increase the adhesibility of any protein by engineering.
Thank you, sir. Actually, I didn't show the study. Already in our group, there is a people, they are working for the chronic wounds. So what they did is the same kind of protein, but they engineered the protein in such a way that it can easily tunable to change the hydrogel behavior, pH changeable, temperature changeable, like kind of behavior. They take the secretome, like for example the cells, correct? Once they grow the cells, and then when the differentiation stage, they just break down the cells and incorporate the molecule, and it is helpful for the chronic wounds. So what we are trying to first establish is, this is the native protein. So working with the native protein is much challenging than the small proteins like peptides. So first we wanted to make the formulation, already formulation has been done. So now we wanted to see whether it can efficiently show as a wound healing material first. Then only we can either, like as you said, vitamin C or whatever material we can incorporate it, that we can easily incorporate and we can tunable, sir. So, but so far what we, we modified the protein, correct? But that is not so far working in our saccharin protein, because the native protein itself, it is not with the help of the dopa molecule. It is because of the hydrogen interaction. Hydrogen interaction only it can interact, because it has a random amorphous domains, correct? So all the tyrosine molecules are present in the hydroxyl, amorphous domain. So that might have an interaction. So that is the reason that engineering does not work in the saccharin case. But when it comes to the dopa molecule, the additions are similar, dopa, but the thing is, in the case of tyrosine, when you incorporate it, it go for aggregation. But in the case of a dopa, it can formulate like a kind of a particle like that. So we can easily load the drug and we can deliver into that. So we also did it, sir. So we load some kind of mRNA based drugs, correct? It can easily bypass the cell wall layer, because I don't know whether based on the lipid layer or something like that. Easily when we do the phase separation molecule used for the drugs, correct? One major challenging for us is, in this case, we have to control the size, because the phase separated molecule over the time, it started to merge and form a larger size particle. Even the larger size particle, it can penetrate inside the cell wall, but the thing is, we have to control it, correct? Controlled manner we have to. So that is what we are working on it. Now we have to control the size and also easily burst out once it is released into the thing. Yes. Yes. Thank you. Yes. From student.
Good afternoon, sir.
Good afternoon.
Sir, regarding the saccharin hydrogels that you had spoken about, the studies that you had taken, it showed that the wound healing was about for a particular period of time.
Yes.
Is it possible to modify it to heal it within minutes to treat hemophilia?
Okay. Yeah, we can block, you mean the healing will not take within a few minutes. Only we can stop the blood bleeding, correct? That we can stop in a minute. That already has been established using different kind of material. But for our aim is not to stop the bleeding. We wanted to go for a healing. Okay? Stopping bleeding will be a different material based on that.
Thank you. And the other thing is the mantis scrap that you had said.
Yes.
So, like you talked about the bulletproof and all of that. So can we integrate it into houses to help people be saved in earthquake-prone areas?
Yes, but the thing is, the challenging part is it is a biomaterial, correct? To make the biomaterials from the recombinant protein, if you are working on a recombinant protein, then it is challenging to produce it. If you liter, for example, 1 liter of culture, if you express the protein, correct, you will get hardly 20 to 30 milligram only. The bottleneck was that. So the production cost will be high in this case. Like maybe what we can try to do is, maybe we can make some kind of a composite material. We can make out of it. That we can do. Or otherwise we can try to find out which peptide is responsible for this phenomena. Or then we can easily produce the peptides, then we can integrate into it. In this, I think from the mantis swim protein, correct, we found out that 2 domains was the calcium binding domains. So that might be the responsible for the hydroxyapatite formation. So yeah.
Students, do you have any other questions? Thank you, sir, for the thought-provoking session.
Thank you once again for your patiently waiting for that. So, thank you.
Now, it's time to visualize the creative findings of our participants through their colorful poster presentations. Others get down for the open-air stadium for their foods. I request the participants to move to their allotted starts. For the respective faculty members, the food is arranged in first BSc.